This is quite an interesting topic for me, but I am more an academician. 2 years ago, I was here speaking about a more mystical topic, which was how Jeffrey Yuen, an integrative approach to male infertility based also on Jeffrey Yuen's teachings. So now I will speak more about more theoretical things which are more relevant now in the research done in, especially in China, in TCM. I would like to thank the organizers for inviting me here. And I would like to know how many of you know what system medicine is? One? Okay, great. Have you ever heard of systems medicine? Never? Hands raised. Who heard about it? Okay, a couple of people. Systems medicine is what the previous speaker, Dr. Loku Gamage, defined as designer medicine. So this is very relevant, both in terms of Western medicine and Chinese medicine. So the World Health Organization, since the 70s, reaffirmed the commitment to the principle enunciated in its constitution, that the enjoyment of the highest attainable standard of health is one of the fundamental rights of every human being. But is this possible? Right now, Western medicine is having a huge crisis because the costs are increasing, and the side effects of the treatments are the 6th cause of hospitalization in the US. And the costs of developing new drugs are huge, and as a consequence, fewer new drugs are released in the market. The cause, one of the causes of all this is because Western medicine is based on a reductionistic approach. That means that it is based on a point-to-point methodology where a single small molecule acts on a single target. So this is especially useful if you're speaking about acute diseases. Actually, this is a model for infectious diseases where antibiotics are targeted against a single germ. And it is also helpful for conditions where there is a single cause. But this is not equally effective for multifactorial complex chronic diseases, as we see mostly today. This is because variability is the law of life. And no two faces are the same, so no two bodies are alike, and no two individuals react alike and behave alike under the abnormal conditions which we know as disease. So what can we do about this? The answer has been found in systems medicine. System medicine was born after the Human Genome Project, which has profoundly transformed biology and medicine by decoding all the information in the DNA. So systems medicine, based on this information, gives us information on how a biological system responds to a given disturbance or stimulant of the environment. In systems medicine, the organism is viewed as an integrated and interactive network of genes. So this is studied by genomics and transcriptomics. Network of proteins are studied by proteomics, and network of biochemical reactions studied by metabolomics. So first, some definitions. The Human Genome Project decoded the DNA. What is the DNA? Do you know what is DNA? Yes? Okay. So basically, it's the hereditary material in humans and almost all other organisms. And the information is stored as a code made up of 4 chemical bases. And just think that human DNA consists of about 3 billion bases. So the structure of DNA is like a ladder. So the base pairs form the ladder's rungs, and there are also other parts that are important which are around the ladder. And the important property of DNA is that it can replicate or make copies of itself, and it can be copied by other structures to make proteins and all what is necessary for a cell to replicate. About 99% of all these 3 billion bases are the same in all people. So we have a strong common base and another part which makes our human unique variability. A gene is a part of a DNA. So DNA is all this huge number of pairs, but some parts of it are what are called genes, which are units, basic units, both physical and functional, capable of expressing and exerting phenotypes or metabolic changes. That means that genes produce proteins which then intervene in the metabolism of the cell or of the organism and express different forms. So if I have brown hair, I have different genes than people who have blonde hair. And in humans, genes vary in size for a few hundred DNA bases to more than 2 million bases. And we have about 20,000 to 25,000 genes. These genes are condensed in chromosomes. So each chromosome is made up of DNA, which is tightly coiled many times around proteins called histones that support its structure. So chromosomes are visible when the cells replicate. And we have 46 chromosomes, and the chimpanzee, how do you say chimpanzee? Chimpanzee have 48. So this is quite a number. So we can think that maybe the more the chromosomes, the more intelligent we are. But also because, for example, the pea plant has 14 chromosomes. So we can say, okay, we have more chromosomes. We are more intelligent than a pea plant. But I don't know if you are cat people or dog people. Cats have 38 chromosomes, but they're also sometimes quite intelligent. However, dogs have 78 chromosomes. So are they more intelligent than we are? Well, sometimes I think yes, but are they more intelligent than cats? So this is up for debate, but basically the last straw is the goldfish. So this means that it's not true that more chromosomes means more intelligent, or who knows, maybe we have to define intelligence. But there is not really a relationship about the number. So to summarize, if we think about a book, well, how does this work? Oh, okay. The sentence, so the words, the letters are the basis. The sentence is the gene. A chapter is a chromosome, which is a series of genes with a function. And the whole book is the genome. So we have difference between genetics and genomics. Genetics is about single genes which codify for single proteins. The genome is the whole book, the study of the whole system. So also the blank pages of the book, the index, every part of the book. So it is quite a huge difference. So genetics It has mostly to do with heredity. Yes. So it examines the function of the single gene. And so the genetic test can look at variations of single genes which code for different proteins. So the statement that genetics is not relevant for disease is because single genes are not relevant for disease only for about 15–20% of the cases. But we are speaking about single genes and mutations. So this is a whole chapter. I talked about it 2 years ago when I spoke about male infertility. There was a whole chapter about genetics in male infertility. And the study has been done on single genes, single receptors. For example, diseases of the androgen receptor can lead to male infertility, but they only explain a small, very small percentage of disease. And what is, how do genes intervene in disease? For example, the correct sentence is the gray cat ran down the hall, and a single mutation of a gene modifies the sentence as the gray cat ran down the ball. So, of course, that doesn't make sense, and there will be disease after this modification. Sometimes, however, the modification of a gene does not lead to disease. For example, if we have, the gray rat that ran down the hall, that still makes sense. So some mutations of genes are not cause of disease, but other mutations are cause of disease. However, the single gene approach has not given much insight into cause of disease. So if genetics is the study of heredity, genomics, on the contrary, is the study of all the genes and their functions. And while this is more yin type of approach, this is a more yang type of approach because it focuses especially on the relationships between genes. Because a gene may not be expressed in the same way after a stimulus, but that is too small to be detected by genetics. But genomics instead studies all the relationships between genes and studies the influence of these relationships on the growth of and the development of the organism, but also on disease. And so, while a genetic test examines a single gene, a genomic test examines the relationships of all the genes. So you can see how complex these relationships are. So this has been possible only after the development of technology to analyze this incredible amount of data. So you see here genes, and here are all the relationships between genes. This is a new way of visualizing these complex relationships, because sometimes one picture is better than 1,000 words, but here really 1,000 words and 1,000 relationships. So from all these relationships, we can see the possible effects of this kind of response on function, regulation, and interactions of gene products, so proteins and other things. For example, we have chromosome 22, which is one of the first chromosomes which has been fully sequenced because it's small. And you can see the genes. So all these P and numbers are, Q and numbers are the genes that have been identified. So you see there are quite few. But look at the interactions of all these genes given different stimulants. So it is a huge amount of information. So this is the difference between genetics and genomics. Is it clear? Yes. Still anybody asleep? No. Okay. So have you heard about these words? Reductionism and holism? So what does it mean? So this is about the concept of system. So reductionism looks at a single piece of the system, and it has given us an incredible amount of benefits, because definitely Western medicine is extremely effective in a variety of situations. However, some parts, is it possible that looking at one part, we can have information on how to heal the whole system? So this is a philosophical but also a very practical question, because looking at the basic parts, sometimes we miss information that can be gathered only looking at the whole system. And so this is where holism is the advantage of holism. Looking at the whole system, we can have more information which comes from the relationship between the parts. So we both need the yin part and the yang part to understand the whole system. So holism is also a very trendy new age term, but has also now a very deep root also in Western medicine, and also Chinese medicine is a holistic approach because it looks at the whole system. So if we look at the atom, we have some information on the properties of matter, which are very fascinating. But the information that is present at this level is not present when we look at the molecule. A molecule will give us more information, or let's say different information, than looking at the atom level alone. And then if we look at the cellular level, a cell, of course, is made of molecules, but we cannot predict the behavior of a cell if we just look at its molecules. And if we look at a human level, we cannot predict its behavior if we just look at the molecules and the cellular information. So this is why we need a more systems approach. Let's say an approach which is more on a systems level, holistic level. Okay, of course, you know the story of the elephant. There was a village with blind people, and they were asked to describe the elephant. So somebody said that it was like a column, somebody said it was like a flap, and because everybody was looking at a different part of the elephant. So it takes a village to tell the whole story. We need to establish relationships between different types of information and levels of information. So we can look at the information from different perspectives. One is looking at the genome. So we can look at the genetic material of one organism. And then, of course, the genes are transcribed into proteins, and the proteins interact with each other in the metabolism. And so here are some definitions. So the transcriptome is the RNA from one cell. So given a stimulant, how is the DNA transcripted into proteins? And what kind of proteins are synthesized in every cell or in a tissue or one organ? And how do these proteins activate the metabolism in the organism and after one specific stimulus? So these are some definitions. But why should we go through all this? Why is this relevant for health? Because, just to give you the sizes. The genome, we have, as we said, 3 million base pairs, and we about 20,000 genes coding from proteins. We have about 100,000 proteins codified by messenger RNA, and we have about 1 million functional proteins. The information is huge. So why do we need to study all this huge information? Because we can use it to identify, treat, and prevent disease. And how can we do that? Because we can find biomarkers. So this is something you have to keep in mind. Biomarkers. So biomarkers is the shortcut, which is very helpful in everything we do. And biomarkers are useful because they are an increasingly essential element of predictive, personalized, and precision medicine. So they can, instead of looking at all that huge amount of information, we can select some parts of that information which are relevant to health and disease and to that particular individual. So you all know about biomarkers already because they can be indicators of normal biological processes, like, for example, the EKG. How do you say here in the UK? EKG or ECG? ECG, sorry. So that's a biomarker. It is some kind of information which summarizes an incredible amount of events that, sorry, what did I do? Of events that go on with every heartbeat. So because there is a pacemaker, here, the P wave, there is a depolarization of the whole ventricles, and there is a repolarization of the ventricles. And all this incredible amount of events can just be summarized with this biomarker. But they are also useful for pathogenic processes. Like, for example, now they have identified a mutation, BRCA1, which, if present in women, it indicates an increased susceptibility to breast cancer. So this is what led Angelina Jolie to have her breasts removed, because she had that kind of mutation. Some biomarkers can also help us determine which individuals will have side effects from medications. And for example, there is this VK or C1, which is a protein, which, if present, determines the susceptibility of people to anticoagulants. So it is used routinely, at least in the United States, to determine what kind of medication for anticoagulation must be used by that particular person. So these are biomarkers which are helpful to decide if a person is healthy or not, and what kind of therapeutic intervention is best. This has been also used with increasing frequency in cancer patients. So systems medicine has helped identify which persons are likely to develop cancer. For example, we spoke about that just right now. And some biomarkers will tell us what type of cancer that is. And it can help determine what kind of treatment is optimal for that kind of cancer in that person, and what is the optimal dose of that treatment. And will that cancer return? So you see all very important questions. And for example, people with cancer can be stratified according to the different mutations, or, well, this is just a nice picture with nice colors, but it's very complex. But specifically in colorectal cancer, patients can be stratified with different subtypes of the tumor, and then the prognosis for each subtype. So all this is very helpful for treatment and for patients. And this kind of information has led to more personalized approaches in Western medicine. So personalized medicine is one of the keywords now. But we know Chinese medicine, and Chinese medicine is a systems medicine because instead of a point-to-point methodology, it has always used a system-to-system methodology because TCM is concerned with the body as a system. And how does it do it? With differential diagnosis, the syndromes. So the syndrome is a set of symptoms of both pathological and physiological states. And we can define the syndrome based on the chief complaint, the sign and symptoms, and the 4 methods of diagnosis. And the differential diagnosis would lead the treatment strategy and the therapeutic prescription. So this is not something which is foreign to us. And so in the reductionistic approach, we have a single drug, one target, one gene, specific therapeutic targets, which target the disease. But in a holistic approach, as Chinese medicine, we have a multi-component synergy with networks of multigene, ligand protein. We will go into that shortly, which lead then to the diagnosis and the differential diagnosis. So we have a different approach because we look a lot at the phenotype and the features of the disease. However, the problem is that it is difficult to be consistent in the diagnosis. If you do TCM diagnosis, you have to look at many things, and there is not much consistency, both between raters, and the same rater can give different evaluations to the same patient depending on many subjective factors. So having a reliable way of diagnosing the TCM patterns is in general difficult. And this also impacts research because stratifying the patients according to the diagnosis is difficult. So probably this is why research has been done in a herd medicine fashion, that is one treatment fits all. But this is not TCM. So, for example, the difficulty of giving a correct diagnosis of the tongue color has been studied a lot, especially in Korea, where they have come with systems, photographic systems with, which rate the colors in attempts to make this more objective. But one simple way to stratify the patients, of course, syndrome differentiation is extremely complex. But it has been simplified now in hot and cold syndrome. So this is a way to start looking at things in a different way. So not one treatment fits all, but let's look at this, let's say, coarse, not really sophisticated diagnostic system, but at least we have some differentiation, some stratification. So we can look at the macroscopic level at the patient, and we can see if the patient has certain characteristics, can be put in the cold column, or if he or she has other characteristics, can be put in the heat column. So all this is very simple. You all know what are the different characteristics of cold. The patient feels cold, is not thirsty, is pale, profuse urination, and the tongue is pale and has a white coating, while in the heat syndrome, the patient feels hot, is thirsty, has a red face, no urine, red tongue, and yellow coating. So these two syndromes can be found in many pathological conditions. And with systems medicine applied to Chinese medicine, we can look what happens at the microscopic level in these two syndromes, because we can look at biomarkers identified by genomics. So this is what research in China in the last years has been looking at to apply the methodics of genomics, proteomics, metabolomics to Chinese medicine to identify biomarkers which can help us identify the syndromes. So it is really fascinating, and I'll try to give you an overview of this. Because at the microscopic level, we can look at the biomarkers. And we can see if, looking at genomics or proteomics, we can see different patterns which segregate with different syndromes. So we can look if at the microscopic level there is a specific imbalance for the hot syndrome and the cold syndrome. And actually, this is true. Otherwise, I wouldn't be here. Speaking about this. So, for example, this is what happens in patients with acute with gastritis. They looked at people with gastritis, and they stratified the patients in hot and cold syndrome. And they looked at the gene expression in those patients. And so you can see the blue points are the genes that are expressed in the cold syndrome, and the red points are the genes expressed in the hot syndrome. And the purple ones are common to both. So you see there is a large part of purple dots, points, circles, how can you say that? But some genes are expressed only in the cold syndrome, and some genes are expressed only in the hot syndrome. So those will be the biomarkers. And so the purpose of all this is to make a model so you can look at the biomarkers. And this is what they have done in the gastritis patients. And so they have seen that in the cold syndrome, the leptin genes were overexpressed. So leptin is an hormone which gives a sense of I have eaten enough. There is, it lowers the nutrition intake. And there were also genes expressed which pointed to the oxidative system. So basically, we know that when you have a cold syndrome, you are slowed down, because this is what cold does to you. It slows you down. And you can see that also at the microscopic level, that there is a low energy metabolism. So also the metabolism is slowed down in the cold syndrome. On the contrary, so here you see leptin levels in the cold syndrome were increased, while in the hot syndrome were not significantly different from normal people. On the contrary, in the hot syndrome, there was an overexpression of genes which were involved in the immune response and immune regulation. So we all can imagine that hot means inflammation. Inflammation is the activation of the immune system. And so you see these biomarkers were more expressed in the hot syndrome than in the cold syndrome compared to the normal population. So there is a correspondence between the macroscopic level. We differentiated the patients in hot and cold syndrome. And this also reflects in the microscopic level in the expression of the genes. So you may say, well, of course, if one is cold, something must happen also in the genetic level. But it is fascinating to see that that correspondence, or maybe I'm the only one who gets excited by this. So the hot and cold syndrome has been studied also in rheumatoid arthritis. You all know what rheumatoid arthritis is because you are physiotherapists. So it is an inflammation of the synovial membrane, which then leads to